Hello dear students. Today we are going to see the introduction about plant growth promoting rhizobacteria and their mechanisms involved into the plant growth promotion and by control of various phytopathogens. So in this lecture we are going to cover the introduction about the various microorganisms present into the rhizosphere for their ability. They are well known for the plant growth promotion and biocontrol. So there are two type of mechanisms which are covered into this lecture for the plant growth promotion, that is the direct mechanism of the plant growth promotion and indirect mechanism of plant growth promotion. Later we will see the commercialization of these rhizobacterias in various uh, crop improvement processes and their advantages. So, first we will see that what are the PGPRs. PGPR क्या है? So these are nothing but a group of soil-borne bacteria which are also known as a rhizobacteria. ये सभी बैक्टीरिया जो है राइजोस्फीयर्स में, लास्ट लेक्चर में मैंने राइजोस्फीयर क्या है ये अच्छे से आपको एक्सप्लेन किया था. So the microorganism present into the rhizosphere are not all the microorganisms which are present into the rhizosphere are known as a rhizobacteria, but the special type of microorganism or the microbes having a special characteristics of plant growth promotion are known as a plant growth promoters or plant growth promoting rhizobacteria. They are actively colonizing around the roots or on the surface of the root into the rhizosphere. By that they enhances the plant growth and ultimately which results into the increase into the crop yield. So they have two mechanisms, direct mechanism and indirect mechanism. The plant growth promoting rhizobacteria were introduced by Joseph Koepler and M. Sloth in 1978. So they are, we can call them as a father of this PGPR term. So here is the profile of Dr. Joseph Koepler, who is from the Auburn University, and he introduced the term plant growth promoting rhizobacteria. So what is the rhizosphere? Initially, we'll see that the rhizosphere nothing but the area around the roots of the plant. So that area is called as the rhizosphere. Generally, 2 to 5% of the bacteria are PGPR, means the microorganism or the microflora present into the rhizosphere around the root. Among them, 2 to 5% microorganisms are of plant growth promoting group. So, we can say that the population present around the roots into the rhizosphere of microbes. Among them 2 to 5% are plant growth promoters. So what are the basic characteristics of this PGPR? First, they should colonize the root, means they should have the habitat or the we can say that they should survive around the roots in the symbiotic kind of relation. So then the survival or multiple in microhabitat associated with the root surface in competition with the microbiota. Obviously, we saw that they are only 2 to 5% of the microorganism which comes under the PGPR group. So they have the competition with the non-PGPR microbes. So they should have the ability to compete with these non-PGPR microfloras or microbes to survive around the roots, as well as they should protect themselves as well as to the plant root from these harmful or disease causing pathogens. So these kind of abilities should these PGPR have. Then they should have the ability of plant growth promotion, and also they should have the plant protection activities, which is also called as a biocontrol. Now we saw that There are two types of PGPRs present on the basis of their presence. If the PGPR is located around the root or on the surface of the root, but not they are not able to get penetrated into the cell. So these kind of microorganisms or PGPRs are called as a extracellular PGPRs. Where they will be present? They will be present into the rhizosphere, rhizoplane. Rhizosphere means in the area around the roots, and rhizoplane is the surface of the root or the space between the cells of the root cortex. But they won't be able to get penetrated and activated into the cells, root cells. And another type of PGPR is the intracellular PGPR. They are able to get uh, penetrated into the root cells, and because of that they forms a nodules onto the root, and these nodules are helps into the nitrogen fixation that we are going to see in the detail later. So basically, we on the basis of the presence of the PGPRs, they are divided into two types, either extracellular if they are into the rhizosphere or rhizoplane, and intracellular if they are able to get penetrated into the cell, root cell, and forms the root nodules. The extracellular type of PGPRs won't be able to form the root nodules. Then on the basis of their functions they are again classified, or on the basis of their functions their activities are classified into two types: direct activity or direct mechanism, or indirect mechanism. In direct mechanism, PGPRs are directly involved into the plant growth promotion or biocontrol, or directly they will be having activity onto the plant growth promotion. So like nutrient cycling, phytostimulation, like that. These are the different types of activities which are going to discuss in detail later. And the indirect activity, in indirect activity they will be producing some metabolites which are indirectly helping the plant into the plant growth promotion or into the protection against the various abiotic stresses. So these are the different mechanisms of the plant growth promoting rhizobacteria. In direct mechanism, the biological nitrogen fixation is there, phosphate solubilization, and phytohormone production. While in indirect mechanism, there are different metabolite production are there, like lytic enzymes like glucanase, chitinase, protease, cellulase. These are the different lytic enzymes which are being produced by the plant growth promoters. Also, along with that, they are having the ability to produce the various antibiotics, and they are able to induce the systemic resistance into the plant. It's just like a immunization of the plant, as well as they are having the ability to produce the ACC desaminase. Whereas the Siderophore molecule, which has been produced by this PGPR, is comes under the both, that is the direct mechanism as well as into the indirect mechanism, because this Siderophore molecule is having the ability or the importance into the plant growth promotion as well as into the biocontrol. So first we will see the direct mechanism. In that we have the first mechanism is the biological nitrogen fixation. So the nitrogen is a very vital nutrient for the plant growth. 78% of the nitrogen is present into the atmosphere, but directly the plants won't be able to utilize that nitrogen themselves. They has to get it in a utilizable form, and the conversion of the atmospheric nitrogen into the utilizable form is done by these rhizospheric bacteria or the PGPRs. So biological nitrogen fixation fixes 60% of the earth's available nitrogen for the plants. If we see into the weightage, so it is a 20 to 30 kg per hectare per year these microorganisms fixes for the plant. So they are the use of these various chemical fertilizers or the supplements for the nitrogen are no need to use these kind of the chemicals. So these rhizospheric bacteria are also referred as a biofertilizer because of their activity into the providing the various fertilizers biologically to the plant. The symbiotic bacteria means we have seen that there are two type of bacteria, intracellular or intracellular or extracellular. So the biological nitrogen fixation comes under the intracellular type of bacteria, and extracellular bacteria. Both are both the type of uh, PGPRs are able to fix the atmospheric nitrogen. First, we will see the symbiotic. The specificity, the infects the root by producing nodules. So, the leguminous plants are and non-leguminous trees having the different type of PGPRs for their biological nitrogen fixation. The leguminous plants are getting nitrogen fixed by the rhizobias, while the non-leguminous trees getting their nitrogen by the frankia. The rhizobias are intracellular PGPRs which are invading the plant's root cells and forming the root nodules. And there they are getting multiplied. It's like a gangrene to the plant root. So there they are getting survived and they are helping the plant by fixing the nitrogen as well as they are getting their shelter there. So it is a kind of symbiotic relationship by which they both are getting benefits. So here is the structure of root nodule. You can get how they are getting or they are infecting the plant root for their survival, and then how they are going to form the root nodule. The another type is the non-symbiotic type of biological fixation. In that the microorganisms are not getting benefited by the plant here, but the plant is going to be get benefited here. So here the free living diazotrophs associatives or non-specific or symbiosis, here is are the different strains of various microbes which are involved into the non-symbiotic type of nitrogen fixation. The azospirillum, azotobacter, buccordia, herbospirillum, bacillus, and penibacillus. These are the different bacteria, PGPRs, who are involved into the non-symbiotic type of biological fixation. So what is this biological fixation? You can see here in the flow chart. Here are the PGPRs which are able to fix the biological nitrogen, atmospheric nitrogen. And here are the various amenophying bacteria which are able to uh, convert the unutilizable organic matters into the utilizable ammonium for the plant, and that ammonia and the atmospheric nitrogen are then uh, transferred in the form of nitrate with the help of nitrifying bacteria to the plant. So this is how the atmospheric nitrogen and the organic materials are converted into the nitrate and provided to the plants. So this is the genetic level of the nitrogen fixation. Here is the NIF gene cluster codes for the nitrogenase enzyme, which is a very important enzyme into the biological nitrogen fixation. And the enzyme has been produced by the various species of PGPRs. So this is the enzyme which is having a main role into the biological nitrogen fixation. This is the another important mechanism of PGPRs after the atmospheric nitrogen fixation, that is the phosphate solubilization. Phosphate is the very important nutrient for the plants, but as it is in a insoluble form and in a rocky form, the plants are not able to utilize that phosphate. So the plants are only able to absorb the mono or dibasic of phosphates. So generally, what the PGPR used to do, PGPR mineralizes organic or inorganic phosphorus in soil, and they converts, or we can say that they converts the insoluble form of phosphate into the soluble form or the utilizable form of the phosphate. That's why the rhizobacteria having the capability of solubilizing the organic or inorganic phosphorus into the soil are also known as a phosphate solubilizing bacteria. The primary mechanism of phosphate solubilization is organic acid secretion by microorganism, utilizes sugar from the root exodes, produces organic acid like acetic acid, lactic acid, malic acid, and so on, act as a good chelator of divalent calcium ions. They accumulates the dibasic or monobasic calcium ions for plants. Then the other mechanisms of these phosphate solubilizers are the produces extracellular enzymes like phytase, hexaphosphate, salts of inositol, that is a phytate, and then phosphophosphatase. So this phosphatase is enzyme again used into the phosphate solubilization. They releases of phosphate during the substrate degradation. This is the mechanism of phosphate solubilization. In that we can see here the rhizospheric soil is here, and in non-rhizospheric soil is here. So in from the non-rhizospheric soil, they solubilizes the phosphorus and converts from the organic or inorganic phosphorus, and after the solubilization, the phosphorus or the phosphate is solubilized and transferred into the rhizospheric soil. And after that, the rhizospheric from the rhizospheric soil, the roots will get absorb that phosphorus. The third important mechanism of PGPR, that is from again from the direct mechanism only, that the phytohormone production. So as we know that as the humans are required various different type of enzymes for their growth and different metabolic activities, like that plants are also required the various growth hormones for their various metabolic activities. Among them, there are three major hormones which are required for their growth and proliferation. So, auxin, gibberellin, and cytokinins are the major hormones which are required required by the plants. And again, there are species which are present into the rhizosphere are able to produce these phytohormones. So first phytohormone is the indole 3 acetic acid, or generally it is called as an auxin, which is involved into the cell elongation, cell division, tissue differentiation, aids apical domains, highly developed roots, uptake more nutrients. If the indole acetic acid is provided to the plant in a good quantity, then the plant will get nicely grown and the plant will be having the maximum productivity. So here is the biosynthesis pathway of indole acetic acid by PGPR. Then the second type of hormone is the cytokines, which are required for the cell division, root development, root hair formation, shoot initiation, inhibition of root elongation, and various things in which the cytokines are involved. So they influence their physiological and developmental processes, and here are the major three species who are able to produce the maximum amount of cytokines are the Pseudomonas, Azospirillum, and Bacillus. Are the three major PGPR species who are able to produce the cytokines. Other PGPRs are also able to produce the cytokines, but these are the major cytokine producers. So here is the again biosynthesis pathway of the cytokines. Then the gibberellins are the phytohormones who are involved into the seed germination, seed elongation, or and flowering and fruit settling. So we can say that this is the hormone which is very essential for the formation of new generation of the plant. That's why these hormones are also very essential into the life cycle of the plant. So there are 136 gibberellins are known from the plants. Only 4 from PGPR, GA1, GA3, GA4, and GA20 are produced by the PGPR. The effect of PGPR producing gibberellic acids on the plant is not exactly known, but when they produces the gibberellins, the growth of the plant has been observed very good in presence of these PGPRs. While the indirect mechanism now, so here we had done with the direct mechanism of the PGPRs. Now we are going to see the indirect mechanism of plant growth promotion or biocontrol by the PGPRs. So first point into the indirect mechanism of plant growth promotion is the Siderophore production. So as we know that iron is an essential nutrient for the growth of the plants, but rather it is very abundant into the nature, but still the plants are not able to utilize these that iron directly from the soil. Again they have to be into the utilizable form, and the conversion of iron into the utilizable form is known as the chelation of the iron molecules, and the chelation is done by the siderophore molecules. So that's why the to for the uptake of the iron molecule from the soil by the plants, siderophore plays a very important role by chelating the iron molecules. Along with that, the siderophore has the another mechanism against the phytopathogens present into the rhizosphere by competing the or by producing the antifungal metabolites. These siderophore molecules inhibits the fungal growth. The release of iron chelating molecules to the rhizosphere improves the iron nutrition, attracts the iron towards the rhizosphere, inhibits the growth of various microorganisms. As I said, that the fungal species or the phytopathogens. Phyto means plant, pathogens mean the disease causing disease causing microorganisms. So these disease causing microorganisms of the plants are generally called as the phytopathogens. So the siderophore molecules helps into the inhibition of cell wall of the phytopathogens. Major two bacterial strains have been identified for the production of siderophore molecules is the Pseudomonas fluorescens and Pseudomonas aeruginosa. So here is the flowchart of the siderophore delivery mechanism. Then the another type of indirect mechanism of plant growth promotion is the production of lytic enzyme. Now, first we will see what are the lytic enzyme. The enzymes which are able to lyse something. ऐसे enzymes जो डिग्रेड करने की एबिलिटी रखते हैं किसी मॉलिक्यूल को कॉम्प्लेक्स फॉर्म से मोनोमेरिक फॉर्म में कन्वर्ट करने की एबिलिटी रखते हैं, these kind of microorganisms are referred as the lytic enzymes. So basically, glucanase, chitinase, cellulase and protease are the important lytic enzymes which are involved into the biocontrol of the phytopathogens. These enzymes directly acts on to the cell wall, and the components of the cell wall are composed of glucan monomer, chitin monomer, and protein monomers. So these monomers are get degraded by the lytic enzymes, and then the fungal cell wall gets degraded, and ultimately the growth gets inhibited. And this is how the lytic enzymes helps into the control of phytopathogens. अब यहाँ पे जो हम केमिकल फंजिसाइड्स का उपयोग करते थे टू कंट्रोल दी फाइटोपैथोजेंस, हमें उनकी ज़रूरत नहीं रहेगी, एंड दिस इज़ हाउ द बायोलॉजिकल सॉल्यूशन हैज़ बीन फाउंड. इससे दो फायदे होंगे कि बायोलॉजिकली उन फाइटोपैथोजेंस का कंट्रोल करने की वजह से हम कोई केमिकल नहीं यूज़ कर रहे, तो वो केमिकल्स जो है हमारे सॉइल में मिक्स नहीं हो गया, और उसकी वजह से होने वाले जो साइड इफ़ेक्ट्स हैं वो कम हो जाएंगे, ख़त्म हो जाएंगे. और उन साइड इफ़ेक्ट्स की वजह से जो हमारी क्रॉप प्रोडक्टिविटी में इफ़ेक्ट हो रहा था, वो इफ़ेक्ट सबसाइड होते जाएगा, ख़त्म होते जाएगा, और वो जो प्रोडक्शन हमारा उस केमिकल की वजह से कम हो रहा था, वो प्रोडक्शन दोबारा रिन्यू हो जाएगा, और इस तरह से हमें 100% प्रोडक्टिविटी मिल पाती है. देन द एंटीबायोटिक प्रोडक्शन, माइक्रोबियल antagonistic antagonist again the plant pathogens, alternative to chemical pesticides. Bacillus and Pseudomonas species are found effective into the antibiotic production. जो फंजिसाइड्स सॉरी पेस्टिसाइड्स हम लोग यूज करते थे टू कंट्रोल द वेरियस पैथोजेंस, उनकी अगेंस्ट ये प्लांट ग्रोथ प्रमोटिंग राइजोबैक्टीरिया जो है एंटीबायोटिक्स का प्रोडक्शन करते हैं. ये एंटीबायोटिक्स उन पेस्ट को कंट्रोल करने का काम करता है, खत्म करने का काम करता है. देन अगेन हियर द सेम, जो पेस्टिसाइड्स हम यूज करते थे उनका अल्टरनेटिव बन के ये प्लांट ग्रोथ प्रमोटिंग राइजोबैक्टीरिया है. तो ये प्लांट ग्रोथ प्रमोटिंग राइजोबैक्टीरियाज का जो उपयोग है, इनका जो एक्शन है, इसकी वजह से हमें पेस्टिसाइड्स का उपयोग करने की ज़रूरत नहीं है. एंड अल्टीमेटली इफ वी लीव द यूज ऑफ दिस पेस्टिसाइड्स, सो द साइड इफेक्ट्स जो उस पेस्टिसाइड्स की वजह से हमारे क्रॉप्स पे होने वाले थे वो नहीं होंगे, और जो प्रोडक्शन हमारा कम हो रहा था उन पेस्टिसाइड्स की वजह से वो नहीं होगा. Then the induced systemic resistance, it protects the plant from the various diseases, increases in the level of basal resistance to the several pathogens stimulate simultaneously. uh, एक तरीके से हम ऐसे बोल सकते हैं कि जैसे हम ह्यूमंस को वैक्सीन देके ह्यूमनाइज करते हैं, या फिर उस स्पेसिफिक पैथोजेंस के अगेंस्ट लड़ने की एक रेजिस्टेंस पावर उन ह्यूमंस में प्रोड्यूस करते हैं, उसी तरह से ये जो माइक्रो ऑर्गेनिज्म है उन क्रॉप्स में कोई स्पेसिफिक पैथोजन के अगेंस्ट लड़ने की शक्ति पैदा करते हैं या ताकत पैदा करते हैं. दिस इज कॉल्ड एज द इंड्यूस सिस्टेमिक रेजिस्टेंस. So the Pseudomonas strain are found effectively to induce the systemic resistance into the various plants. So here you can see the mechanism of generation of induced systemic resistance. Then the plant stress marker production from the environmental stresses like temperature, cold, drought, salinity, alkalinity, UV radiation, pathogen infection. These kind of environmental stresses can be indicated by the plant promoting rhizobacteria. So Then this is the important one, the production of ACC desaminase. So the ACC stands for 1-aminocyclopropane 1-carboxylic acid, which is the precursor of phytohormone ethylene. It increases dramatically under the abiotic stress. It has the demerental effect on plant. If the ACC desaminase is not getting produced, your ethylene enzyme or ethylene hormone will get produced in maximum amount. And ethylene is the enzyme, a phytohormone involved into the fruit ripening. फल पकता जो है वो पकने की जो प्रोसेस है वो इस इथिलीन की वजह से होती है. So basically, the ethylene is produced by the initiation of ACC desaminase. So here is the summary of plant growth mechanism of plant growth promoting rhizobacterias. So you can see various mechanisms: direct, indirect, and combo. In last lecture, I already explained. So there are different plant growth promoting rhizobacterias reported for their various activities and mechanisms for the biofertilization into the biofertilization, phosphate solubilization, siderophore production, exopolysaccharide production, biofixation of atmospheric nitrogen, are different mechanisms have been reported by various researchers timely for the various purposes. The last one you can see, so see here, this is the biofertilizer. Mean these group is used for the plant growth promotion, while the last one you can see here is the control for the control of pathogens, in which you can see that these mechanisms are involved into the controlling the phytopathogens. So when the these PGPRs are commercialized, marketalized और उनको बेचा जाता है दो फॉर्म में. पहला होता है बायोफर्टिलाइजर एंड द सेकंड वन इज अ बायोकंट्रोल एजेंट. सो बायोफर्टिलाइजर इज अल्टरनेटिव टू योर chemical fertilizers, while the biocontrol agents are the alternative to your commercially and chemically available fungicides and pesticides. Several PGPR bacterial strains are commonly available into the dry powder product, suspension form, or liquid form. These are the commercial strains of PGPR which have been used as a biofertilizer as well as biocontrol agent. So Bacillus liqueformis SB3086 and Azospirillum brasiliensis is used for the as a biofertilizer, while Pseudomonas, Streptomycin, and Bacillus species are used for the biocontrol agent. Here you can see that the strain of the Bacillus is used in both the activities, biofertilizer as well as biocontrol. So the strain having the ability of plant growth promotion as well as the antagonistic activity against the various phytopathogens. So here is the conclusion for the today's lecture is the essential of sustainable agriculture. For that we need to go for the biological solutions. Many identified not been commercialized efficiently. Means there are various microorganisms present into the rhizosphere. Among them, many number of microorganisms are already identified, but they are not getting commercialized. So for that also we have to work for. Should do further research on a detailed understanding on their mechanisms. वो माइक्रो ऑर्गेनिजम्स जो हैं ये अब तक जितने मैकेनिज्म स्टडी किए गए हैं उतने ही मैकेनिज्म्स के बारे में हम पढ़ रहे हैं, लेकिन और भी मैकेनिज्म्स उसके हो सकते हैं. सो इट इज essential to study all the different aspects of the PGPR. A lot of effort is still required to make PGPR an efficient technique for the sustainable agriculture. Thank you very much. See you in next lecture.